everyone. Thank you for coming by for my talk. So when we think about coral reef ecosystems and we talk about environmental impacts, we often think about big things like climate change and how this is causing problems for reefs. But there's a lot of smaller impacts that happen environmentally, such as changes in temperature and light, which occur seasonally that can also change and affect coral physiology. And for the purpose of this project, we're talking about this kind of in relation to seasonality of phenotypes, which I might also call coral seasonal phenotypes, depending on how nervous I am. So our symbiont cell densities have been known and fluctuating, especially in Caribbean Florida corals. This is from Fit et al. 2000. You can see there's a natural oscillation in the millions of cells that are kept within the coral's tissues across both these species through time. What's important to note is that we do see higher cell densities occurring in late spring, and we see lower cell densities occurring in late summer. We see the same kind of oscillation with other things that are associated with coral physiology, like chlorophyll pigmentation, which also tends to be higher in late spring and lower in late summer, and the tissue biomass of the whole coral holobiont, which has that same trend. So this kind of led me to more questions. Our lab focuses a lot on physiology, and we're kind of interested in how restoration corals are also impacted by these seasonal differences in phenotype, or if they don't experience that at all because they're in more regulated systems, especially in land-based nurseries. And then we're also interested in how that compares to local wild conspecifics, because we are seeing that in a lot of the wild habitat or wild corals. To answer these questions, we paired up with Mote Marine Labs, Elizabeth Moore Center for International Coral Reef Research. They have a lot of tanks, I'm sure y'all are aware. Lots of beautiful fragments, which they were able to offer us some for seasonal sampling. We looked at two different coral species from their tanks, Acropora palmata and Orbicella fabulata. So these are both framework coral species that are commonly found throughout the Caribbean and the Florida Keys. We sampled 6 separate genets of each of these at late spring and late summer to get those maximal physiological measures. And we're looking at non-destructive measurements. So these are the ones that are commonly done because they don't affect the corals too much. They include things like gross photosynthesis, dark respiration, and FV over FM, which is looking at the electron transport chain in relation to the light reactions of photosystem 2. We also did destructive measurements, which looked at symbiont community analyses. We had symbiont cell densities, chlorophyll A within symbiont cells. So we're kind of using symbionts as a proxy there. And then we looked at total biomass of the host. We then compared these physiological measures to a long-term data set that's also based on the Florida Keys. It's also looking at coral physiology. We used 23 years from this data set, from 1996 to 2019. And this included data from Acropora palmata from 1 shallow reef site and Orbicella fabulata from 3 shallow reef sites, I will note we did exclude data from years where there were environmental impacts. This includes things such as hurricanes and heat waves and even cold spells, because we did want to compare our physiological data at the land-based nursery to what was happening in unstressed wild individuals. Starting off with our symbiont community analyses, these were completed with qPCR measurements. Just to orient you a little bit, we have our 2 species by time point on the bottom, and along that y-axis is going to be our relative percent symbiont community. And these are just the Mote land-based nursery corals. For O. fabulata, we didn't see really any fluctuation in the symbiont community over time, though we did see dominance with the Durastinium trenchi symbiont, which was typed out with Sanger sequencing. This is interesting because usually these corals in the wild host mixed assemblages that are dominated by a Bervolium symbiont, so these are very different than what we see in those wild populations. For the Acropora palmata outplats, we also saw high Durastinium trenchi associations, but even more interestingly, we saw this dominance by a Cladocopium symbiont, which did type out to be Cladocopium gorui. These are highly different than what we see with Acropora palmata in the wild, though, which almost homogeneously, exclusively associates with Symbiodinium fidi. So that's a whole separate genera, and these are not observed as far as we can tell in the literature so far for these species. Moving on to our photophysiology, we do have a little poorly done this is the best picture I had of our respirometry chambers. Please ignore the Cassiopeia that are in there. So we have a graph here showing our gross photosynthesis to dark respiration ratio. And then up on the bottom, we have palmata and ovabulata. The blue, sorry, I cut off our figure legend, but the blue here is spring, and the gray is our late summer sampling point. And so you can see for both species, they do have higher photosynthesis generally occurring in late spring than they're experiencing in late summer. Next, we have our FV over FM measurements. So we did do that with a diving PAM2. It's really fancy. You should get one. So we have, I'm just going to orient you this whole figure on the bottom. The rest of them are going to kind of look like this. The red or pink color, depending on how you see it, is going to be our nursery corals in all figures. The blue color is going to be our wild conspecifics. And then they're always going to be paired species by time points for the rest of the figures. So for our FV over FM, like I said, this is looking more at how photosynthesis is working within the host. We did see significant differences across our wild conspecifics and our nursery corals. We're not too worried about this though, because the main reason we did this measure was to assess whether or not they were stressed in the tank systems. And since they're pairing closely enough, we will generally just observe that these guys aren't seemingly stressed in these systems. We then looked at the symbiont cell densities. So once again, on that y-axis is going to be our millions of cells per area in the host. With our land-based nursery corals, we do see higher for both species. A lot of these trends are more obvious in the ovabulata fragments, but we do see higher trends in spring for cell densities being much higher than we're seeing in summer, which kind of makes sense if you think about how corals work. What was kind of interesting, though, is that these were significantly lower than what we observed at all time points for the wild conspecifics, which seems to me indicating that things are a little bit different in the nursery tank systems than they are in the wild. Not too surprising. Next, we looked at our chlorophyll concentrations. So here we have the pigment of chlorophyll, picograms of chlorophyll per symbiont cell. It's important we did it by cell instead of area. And we see an opposite trend of what was expected. So we talked about in the beginning, usually chlorophyll is higher in the winter or springtime than it is in the summer, and the land-based nursery corals seem to be showing higher concentrations of this chlorophyll pigmentation in the summer. And not only that, but those are significantly higher than what are observed in the wild conspecifics for this species. Finally, we looked at tissue biomass. This was in the form of ash-free dry weight per area for these corals. And we have, apomata tends to be kind of thin-skinned, so it has much lower biomass than what we're seeing in the ovabulata, but there is still a trend of the expected higher biomass in late spring than what we're seeing in late summer. The downside of that is that all of measures of the land-based nursery corals were significantly lower than what we're seeing in the wild conspecifics. So kind of just to review what we found, the fluorometry and respirometry data seem to support that these corals, at least with non-destructive measures, are generally healthy. They're photosynthesizing similarly to what can be found in the literature for these wild populations of these corals, and they're not looking very stressed. But our destructive measures of the Symbiodinaceae communities, they were much different than what we see in wild conspecifics. The biomass was really low for these corals, which is problematic, and the Symbiodinaceae communities, though very low, had a lot of pigmentation, which seems to indicate there's low light acclimation occurring there. I'm going to finish way early, I'm sorry. Conclusions. So our final conclusions, those first questions I asked you. Yes, we're seeing that the nursery-based corals are experiencing some seasonality. Some of the trends we expected are inverted, likely because of low light acclimation that might be occurring there. But the low light acclimation is likely because they're experiencing a quarter of the light that the reef corals are experiencing year-round, in both nearshore and offshore habitats of the Florida Keys. And this could mean that high light becomes a source of stress during acclimation of outplants to these reef habitats, which would mean a lot of energetically costly transformations both within the host and the symbiont cells to adjust to those new environments. And this is kind of problematic because of the low biomass that was measured. So with low biomass, you have lower energy reserves to use for these kinds of transformations, which includes lower competitive advantage for those corals spatially on reef. They have a harder time competing with other organisms and also dealing with predation. And it also means they have less energy available for dealing with environmental stressors such as bleaching events, cold spells, hurricanes, and disease stress. So a lot of you are probably wondering, we talked a lot about the tank. What does this mean when we outplant these corals to the environment? Because that's what's really important. We actually just finished sampling a project last week, right before the hurricane, using A. palmata and O. fabulata. So the same two species. It was a 1-year outplanting. We outplanted them to a nearshore and an offshore reef and kept some individuals in the nursery for a direct comparison around the same age. The sites that we chose were Newfound Harbor and Luke Key. So these are 2 sites that have been highlighted as major restoration sites for the future. So we hope this is helpful for that. And we're going to do the same destructive and non-destructive sampling with the addition of likely lipid class analyses and hopefully RNA-seq if I have time in my dissertation. Maybe not, that'll be a couple years. So we're hoping this gets more into how acclimation is occurring at those sites with these fragments. And at that point, I'd like to say if you have any other questions and I'm not around, my email's at the top. I'd like to thank the Moat Summerlin Key staff and interns and our undergrads because they're wonderful and they were super essential for collecting the data that we got, and the National Marine Sanctuary for giving us the permits and the ability to do this project. And that's it. Thank you, guys.
We time for a couple of questions.
Like not really a question, but maybe a spoiler alert. We don't feed our corals.
Oh.
In our.
We were told y'all did.
In the restoration side.
Yeah,
Okay.
they gave us measures of how much they add to the flames.
Because I was going to say not all of them get fed, and I was like maybe that's why you have a little viral mass.
Yeah.
Okay, so that I might be incorrect.
Can I wanted to ask a question related to this actually. And have you tried validating that light is the driver by just sampling some deeper colonies on the reef where there's less light and see if you replicate the physiology?
I haven't. The data set we have is like a 30-year data set of like the same 6 corals for each species on the reef.
But you could just compare shallow and deep and just see if there's something you can dial out.
I could maybe try. Yeah.
So you figured out that there's a light deficit for the corals coming out of the nursery, and it makes me think back to, well, the way we finish cows and pigs is you feed them a whole bunch right before you're about to put them out. Is that something that you've looked at to hit them with a ton of UV and extra light for the week prior before you're about to outplant them?
Yeah, I think people have seen success with that actually when they move them to, what do we call them, in situ nurseries. Before you outplant, there tends to be more success. That way they get like pre-stress and they have a chance to partially adapt before they have to compete for space on the reef. I think that would be an optimal idea. I don't know if they're actually doing that in the nurseries though, because I'm actually university-based, but. Yes.
So we have a large number of projects, obviously, that have been discussed that use mesoplasms, where you have corals that are in a tank to do some sort of experiment. It seems like from what you showed that those corals may be dramatically different from what we're seeing from their wild conspecifics. How much validity do you think we can give those results, given that those corals are in an entirely different environment?
I mean, I don't know. Corals are different kind of in every environment. Sometimes it's even hard to compare reefs. So I think as long as you're able to get them up to snuff before we start that. So I don't know. Maybe we need ranges of what's a healthy coral versus what's less healthy before we start doing those kinds of experiments so that they're more comparable. I also know that our symbiont associations, the same coral frags were sent to an aquarium and they actually have